Hi everyone, this is Janie Helms, and we are going to be talking about processes that involve glucose. And I'm going to focus on specific processes, but let's take a look at what those are. So these are going to be the things that the presentation is going to cover. This is what we're going to learn, hopefully within that 25-minute time frame. And I definitely will not be going into extreme detail, but just to give an overview of certain things. So the first thing that we're going to cover is we're going to understand the process of glycolysis and gluconeogenesis. Both involve glucose. And then we're going to talk about how an enzyme, the type of enzyme aldolase, is involved in both of them. We're going to talk about all the different isoforms of that enzyme and their expression patterns in the human body. And then I'm going to dive more specifically into one of those isoforms, specifically the Aldolase B, and how it is regulated. Before I go into these processes here, though, I first want to say that the title of this presentation was Processes that Involve Glucose. And I'm sure you know, but I want to remind everybody that we know the importance of glucose. It is a good fuel for the body. It has a very large amount of potential energy within it. It's usually stored in the body as starch or glycogen, and it can be released from inside the cell for the storage so that it can be used for energy production. But I want to give you an overview of these steps and how they connect to everything. So let's talk about the first one, glycolysis. So what is it? In the whole process of cellular respiration, this is the very first stage. The shortened definition is the process that breaks down glucose. It's going to take that glucose and it's going to change it into something that we can use, something that is present in our body. There's two phases, but altogether between the two phases, there are 10 steps. Okay, and there are 5 steps. Do a little writing here. There are 5 steps in phase 1 and 5 steps in phase 2. So equally splitting. The first phase talks about the energy that it takes, and then phase 2 is the energy that is released because of that reaction that breaks down the glucose. So if I want to give you, trying to put the entire process to give it as an overview. Again, there are 10 steps to this process, but I'm not going to go through every little bitty tiny step. I'm trying to give you an overview of the process of what happens. So I'll get my pen back out here, and let's choose a different color. Let's go with red here. So in the whole process, it's going to take the glucose from your body, like I said, and it talks about how I am going to break it down. But it's more involved than that. There's more steps. So basically, glucose, in the end, turns into 2 pyruvates. And then we have the NADH here that you're going to go through a different part of the reaction, and it kind of houses the result or yields NADH plus 2 hydrogen ions. And then I have ADP and phosphate, and they are going to result in ATP and 2 water molecules. Now, in the overall process, if I wanted to give you the breakdown of it, you have, technically it's not written here, but it does start with 2 ATPs, okay? It uses 2 ATPs to start the process because I need energy requiring. I need energy to be able to make all of this happen. And what happens is glucose, it has 6 carbons, okay? It is a 6-carbon molecule. And what happens is that energy is used to take it, and these 2 pyruvates are 3 carbon. So I take 1 6-carbon and split it into 2 3 carbons. And so with that, what comes out of it? I take 2 ATPs, I come out with 2 NADHs, as well as the 2 pyruvates. So even though we're saying that the process is the breaking down of glucose, you can see that there is a lot of different parts, which would explain why there's 10 steps between 2 phases. So let's take it one step further, or let's keep summarizing, I should say. So again, here is our formula, our chemical equation, our reaction that shows what happens. So overall, the 3 chemical transformations is what I previously mentioned. 1, it breaks this carbon. Again, it is a 6 carbon molecule, and it breaks it down into 3 carbon molecules. So 2 3-carbon molecules. Okay. Then you get the phosphorylation of the ADP to ATP. And actually, I do want to go back. Get this here. Oh, let's do this. So glucose to the 2 pyruvates. We have 6 carbon to 2 of the 3 carbon. That's number 1. Number 2, NAD, you have 2 of them. And what happens is the NAD bonds and reacts with electrons. It encounters electrons, and it causes this split. You get this result of 1 hydrogen bonding to the NAD, and you end up getting 2 molecules out of that, plus 2 hydrogens that have separated. Now, when with those 2 hydrogens and with the electrons, it balances out from being a positive ion to a neutral molecule. So again, this is the phosphorylation, or this is the transfer of a hydrogen ion to the NAD. So this is technically number 3 right here. So number 3. And then you have the phosphorylation of ADT to ATP. Now, I said that ATP is used. That's the energy that makes this possible. But I don't have it written in here. That's just what I'm using to make this all possible. But what it actually does, what that ATP does, is it takes ADP and these 2 phosphates here, and when it goes through that reaction, goes through phosphorylation, you actually produce 4 ATPs. So you're probably thinking, why is there 2 over here then? Well, if I use 2 of them and I get 4 out, if I lose 2 because I use 2, okay, I use 2, which means I'm losing. So when I say the 4, the net that comes out of it is a positive 2. So in this formula, even though technically I get 4 ATPs out of this reaction, in the formula to allow it to be balanced, the net that comes out is actually only 2. So again, phosphorylation of ADP to ATP, but don't forget that ATP has to be used in here to make this whole process be able to actually occur. So, glycolysis, to break it down, it breaks down glucose into other forms so that we can use it in our body. Okay, so learning goals. Understanding glycolysis. So that takes glucose and to turn it into something else we can use. Let's talk about the gluconeogenesis, which also deals with glucose, but almost in an opposite effect. So what is it? The overall goal of it, the importance of it, the function, is that it's going to help us maintain our blood sugar levels in the body. Now glucose, again, it is a sugar molecule, which is why we say that this is a process involving glucose. The process actually is inside of our cells. It's going to begin in that mitochondria because it uses ATP, but also in the cytoplasm of the cells. Again, it does deal with glucose. It does deal with energy. So it has similar steps as glycolysis. It seems to some people that it's just glycolysis in reverse, because what it does is it takes those pyruvates, or it takes those substances and turns it into glucose. So glycolysis took glucose, changed it into something else. This process makes glucose. So it wants to store, because as you're through your body, you use up those sugar, you use up that energy in your body, and so that glucose amount is insufficient, and your body needs more. So it takes these pyruvates, those 3 and 4 carbon compounds, specifically I just mentioned 3 compounds, those pyruvates, and turn it into the glucose. It kind of is the reverse process. Instead of splitting 6 and becoming 3 carbons, I have my 3 carbons and now becomes 6 again. So here's a image to kind of show you the breakdown of what exactly it does. And again, this is an overview. There are very complicated. I could go into the structures and the giving and the taking with all of these reactions, but I do want to just give the overview. So humans, we are mammals, we are animals. Most of our tissues, they depend completely on glucose, that energy that it provides. But if I am exercising, going just even the time in between meals, if I go through the long fasting periods, or as I said before with the exercising, that uses up that energy in your body. It uses up the glucose that is stored. So what it does is the glucose that I lose through, whether you want to say you're going through a starvation stage, the fasting, the exercise, what happens is that it's going to take 3 carbon molecules like lactate, glycerol, those pyruvates, and I'm going to talk about what those pyruvates are in a minute. I have them further, some examples, but let's just say pyruvates. It also includes some types of amino acids. So what you're going to do is your body takes these types of 3 carbon molecules and they process them through the liver, your kidneys, your small intestines, the muscle, and even the brain. It takes all of those through there. There's an enzyme that helps, the aldolase. It's going to take those and it's going to put it back together basically into glucose. And the blood delivers that. The blood passes through, the glucose is in the blood and it passes through all of these other organs. So, glycolysis. It goes from glucose to 3 carbon molecules like the pyruvates. Gluconeogenesis takes those 3 carbon molecules and turns it to the 6 carbon molecule glucose. I know it seems like they are just exact opposites, and with these overviews, it definitely sounds like it. But again, it's an overview. If I were to break down every little bitty step with these two, they just share those similar steps I'm saying in the overview. They're not identical. So let's talk about that enzyme that makes these reactions possible. So what is aldolase? Well, how is this involved? There are 2 classes. Okay, it is a type of enzyme, and there's 2 classes. You have class 1 and class 2. I'm going to focus on class 1 because I want to talk about what is going on in humans, in mammals. Okay, but I do want you to realize that there are 2 different classes, and I want you to be aware of where they are found. So, aldolase, it is an enzyme. It is the enzyme that takes the glucose and breaks it up. Here, I say that it can break up the glucose and become fructose. The reason here where it says the aldolase breaks hexose, Hex, as in like a hexagon, 6 sides. Remember that I said that glucose is a 6-carbon molecule. And so it takes the 6 carbon, the hexose molecule, and turns it into 2 of the triose, meaning 3 carbon phosphates. And again, I'm getting ready to say what those phosphates are, but if you do want to know them ahead of time, it is the G3P as well as DHAP. But again, I will say that again, and you'll be able to see a visual on the next slide. But anyway, so the enzyme is what allows that breakup. The enzyme is what allows that the glucose to turn into those 2 triphosphates. And then again, the gluconeogenesis, it's the same enzyme, Aldolase enzyme. It is the part that they share. It is that similarity that seems like they are the perfect thing in reverse. But in the case of this enzyme and the process, it is the process in reverse. It is the same enzyme that is performed in both. So, the 2 processes, how aldolase is involved in both, we know that it is the enzyme that allows these processes to occur. So let's talk about the different types of this enzyme and their different expression patterns. Okay, there are 3. There is, and they just say A, B, C. Aldolase A, Aldolase B, Aldolase C. So I'm going to do each one of them one at a time. Aldolase A, it is located on chromosome 16, and where you find it expressed, it is sporadically involved in most actual tissues. However, its largest expression, it is most extensively expressed in the muscle. The muscle is what it is known for. We know that it has a very crucial role in human physiology. Again, muscles. Muscles, they require that energy, glucose, the sugar, the pyruvates that come out of those processes that I talked about. Not every part of it is completely clear exactly how and why. However, there are obvious results. There's evidence that shows what happens when that enzyme is not working properly. If there's a mutation in the Aldolase A gene, if there's a mutation with it, several diseases are known because of it. This is just one example. It's the NSHA. It is a type of anemia that can occur if there is a mutation with this enzyme. So what it does, Aldolase A is very present in glycolysis. Again, we know that aldolase, I said it was the enzyme, but remember I said we have 3 types. So A is what changes that fructose, or the glucose, to fructose, and it changes it to the glycerol, which is a 3-phosphate. That's why I said one of those triphosphates is G3P. That is the glycerol 3-phosphate. And then you have the DHAP, another triose phosphate, and it's called dihydroxyacetone. And again, we know hydrolysis, or the glycolysis, we said that process results in 2 pyruvates, but we also said that it resulted in water. We got hydro in this word. Di, we got 2 pyruvates. But Aldolase A is extremely important with humans and how our bodies can break that glucose down into these 2 pyruvates or 2 triphosphates. Aldolase B. It's located on chromosome 9. It is expressed extensively in the kidneys and liver, but in very smaller portions in the small intestines. But I wanted to do the extensively, that's the kidneys and liver. If there are problems with this, their main role, so I said Aldolase A was for glycolysis. Aldolase B is for the gluconeogenesis. So that opposite that takes those pyruvates to turn into the glucose, come back. And if it can't do that, those are people that can suffer from hypoglycemia. If it's not working properly, that means that they could suffer from liver failure. The kidneys will have some kind of dysfunction. It could even put the body into a state of shock, and you can go into coma and even die if this is not treated. Your body must have glucose. It must have that energy to function. And if it can't, that means your kidneys and liver can't function, which means your body cannot do what it needs to do. Some signs or early signs could result from the abdominal pain, especially because you have your kidney and liver that is not functioning properly, but there could also be vomiting being experienced by the patient. Aldolase C. That one, we know that it's located on chromosome 17. We know that it is basically anything to do with the nervous system. So it's going to be extensively in the brain, nervous tissues, and smooth muscles like the trachea or the heart. All the research that I found, there is not a hardened fact that we know exactly what its specific role is. However, there is a pattern to where it's found. It is always found in those tissues that have constant metabolic activity. Think about it. Your nervous system, your heart, your brain, it is always having to function in some way. There is always metabolic activity occurring. So we don't know that exact importance of Aldolase C. Why is this enzyme, what is its very specific role? But we know it has to have something to do with that activity that is always occurring. And we know that it is an enzyme that deals with energy and deals with glucose. So we can see that connection. They just haven't fully proven or finalized those beliefs. We know that in the brain, it's a part of the S100 protein binding property. It's also been identified in the zebrin-2, which is an intracellular antigen that helps kind of organize the cerebellum. It is also identified in the inositol triphosphate binding protein that's in our trachea. So, and again, the trachea is another one of those smooth muscles. And we do know that the way that it functions, we believe that it has more process, or it's more closely related to the the Aldolase A instead of the B. So we also think that its properties and its behavior is intermediate with A and B. It's kind of on its own. It's on its own schedule. It's on its own thing. It's not directly related to A or B, but it can be put in place of them, especially since we said that it is more similar to A than B. There's still a lot more to be known about this isoform of this enzyme. So it is definitely one that is still under a lot of study and research. Okay, so glycolysis, turning glucose, breaking it down into two other forms of 3 carbon molecules. The gluconeogenesis, taking those 3 carbon molecules and connecting them back together to create glucose. Aldolase is the enzyme, and that makes both of these processes possible. And there are 3 forms of it, A, B, and C. Now I want to focus specifically on B. There's a lot of confusion that people get, I feel like, from this. Especially with the transcription factors that are involved. So let's talk about a few of them. First of all, the transcription of aldolase B, this gene is stimulated by the glucose and the insulin in our body. And it has been studied that if someone is introduced to fructose, another type of sugar, that it causes the B gene to rise. Basically, the messenger RNA levels, they increase when introduced with that sugar. So there are several regulation factors, transcription factors that control the start side of transcription. There are about 5 elements with respect to how they cause these to happen. The first one that I want to mention is HNF1 and DBP. They are very strong activators to make this gene active and to do its job. Now, HNF stands for hepatocyte nuclear factor. Okay, hepatocyte nuclear factor. Now, a hepatocyte, I probably should write that down here. Let's get that down as a visual. HEPATOCYTE. Okay, this is a type of cell. It is a major cell found in the liver. Now remember when I talked about where this specific enzyme was expressed, it was expressed mainly in the kidneys and liver. So this is a major cell in the liver, and what it does is it activates the immunity against invading microorganisms. The immunity in our body to fight against these things that are invading our cells, this is an important part that helps activate our immunity. So this is one element, as well as DBP, and it helps activate these immunity promoters. Let's exit here. Okay, 2 more, well I should say 1.5 more. You have the EBP, which is a enhancer binding protein. But there's also a variant of this hepatocyte. They are still activators. They still activate our system that release those immunities, but they're a little weaker. Now, the great thing about the enhancer binding protein is that it can actually be strong instead of weak if this other DBP, which stands for just a D element binding protein, if it is inactive, if it's not doing its thing here, it's not performing right, that this can take over and it becomes a strong activator. They think that depends, through all the research they've done, they think that depends on the distal element, or DE, that's bound to the binding sites. So we have a cell in the liver that activates immunity, but then you have HNF3, which is a strong inhibitor of this. And what's really interesting is they actually, the 1 and the 3 overlap their binding sites. So for this to be able to work, you'd have to have these other variants, these other elements that can help still allow this to be activated. So again, all the transcription of these genes, allowing them to do their job, allowing them to activate our immunity against these invading microorganisms, we have to have glucose for that to occur. We have to have glucose and insulin, and we know that that involves, your kidneys and your liver and those processes that involve, which is why that connects to the aldolase B, because that is where it is expressed in large amounts. So if we didn't have the enhancer binding protein and the D element binding protein, these things would not happen, because sometimes this FN or H and F 1 and 3, since they overlap, this is an inhibitor while this is an activator. They could be fighting over each other, back and forth, back and forth. So without these other elements, our body wouldn't be able to do what it needed to do to fight off foreign bodies trying to attack us. So let's go back to this main page. Again, this presentation covered the processes that involve glucose in our body. Glycolysis, glucose gets broken down. Gluconeogenesis, it creates glucose using other things in our body. Aldolase, in all of its different forms, is the enzyme that allows these two processes to happen in different organs, different parts of our body. And probably B, A and B are one of the most important ones, but B has a lot of stuff going on because it is regulated by all these different transcription factors. Okay, so now I can just go through this back to the end here. There's the A, B, C, the regulation, and here are my references. I hope that we got a good overview and a better understanding of these two processes and the enzyme that makes all of this possible and the importance of it in our body. Thank you.